by the Dean of Sciences from both the sides, Dr. Noor Jahan and Dr. Ram Narayan ji. And they had explained the concept and theme of the entire workshop, that what is exactly sustainability and how life sciences can be related to sustainability. Then we had two sessions, where the first session was headed by Dr. Bharti, Assistant Professor, Head and GNC Biotechnology. And she had spoken about the clean city concept and how we can talk about sustainability and a city setup with sustainability. Second session was handled by a very expert resource person, Dr. Vandana ma'am, Professor Vandana ma'am, and she had spoken about women health and folic acid. And she in detail explained the effect of folic and how it works and how it is important for not only women but as such in general. Today is the second day of our session. Today we have our two resource people. The first session would be headed by Dr. Sudhir Upadhyay ji, Assistant Professor, Environmental Sciences, Veer Bahadur Singh, Purvanchal University. Welcome, sir. And the second session would be headed by Dr. Lavanya, Assistant Professor, Biotechnology, Guru Nanak College. Both the sessions would go back to back, and then we will open up for questions. So participants are requested to keep posting their questions in the chat box. Once we open up the floor, you can directly interact with the team also. Another thing is, we would make a request to the resource people that please keep the entire session in English, because there are people from the south who cannot understand Hindi, and there are people from the north who cannot understand Tamil. So we will keep the entire session, keeping in mind the national level, we will keep it all in English so that the understanding is there. And because yesterday we had a few this thing where we were told that the students found it a little difficult when it went into Hindi. So this is a request from the participants that please resource people are requested to keep it in English, which is a common communication language right now here. So Satish sir, can we go live, sir? Thank you, sir. So I welcome all of you to the second day of our National Online Life Sciences Workshop. I request Ms. Shweta Singh, who is a research scholar with biotechnology, to introduce our first resource person of the day. Shweta?
Okay. Can you hear me, ma'am?
Yes, beta, we can hear you. Go ahead, please.
Good afternoon, all of you. It's a proud privilege to introduce Dr. Sudhir Kumar Upadhyay as a resource person of the first session of the second day of workshop. Since September 2010, Dr. Sudhir Kumar Upadhyay has been employed as an assistant professor in the Department of Environmental Science at VBSPU, Purvanchal University, Jaunpur. Dr. Upadhyay has more than 11 years of postgraduate teaching and research experience. He is the erudite scholar and a diligent researcher, work in different CSIR and ICR laboratories, such as National Environmental Engineering Research Institute, Nagpur, National Bureau of Agriculturally Important Microorganism, Mau, National Chemical Laboratory, Pune, Maharashtra, Maharana Pratap University of Agriculture and Technology, Udaipur. The working area of Dr. Upadhyay are microbial ecology, PGPR, plant microbe interaction, eco-friendly amendment for management of degraded soil, environmental pollution management and resource ecology. Dr. Upadhyay obtained his PhD degree in environmental science for Bhim Rao Ambedkar University, a central university, Lucknow, India in 2010 under the supervision of late Dr. Professor D.P. Singh. Dr. Upadhyay secured university gold medal at PG level and also received 4 times Young Scientist Award at different national and international scientific platforms. He is a member of numerous societies such as International Society for the Development and Sustainability, Japan, life member of Greece and the Peace Welfare Society, Academy of Environmental Biology, National Bureau of Agriculturally Important Microorganism, Uttar Pradesh, and the Association for Science and Technology Advancement. Dr. Upadhyay currently working as editor of international journal like a Current Microbiology, Springer of United States, Restoration Ecology, Wiley of Great Britain, UK. Frontier in Microbiology, etc. He is the review of numerous journals such as Environmental Pollution, Chemosphere, Environmental Research, Plant Science of Total Environment, etc. He has published 45 research articles with high impact factor in the Journal of International Repute and has a more than 1,260 Google citation. Dr. Upadhyay attained more than 40 conference, workshop, symposium, training program, and presented paper, delivered talk. He received many times Best Paper Presentation Award. Recently, in 2021, he's received first prize for Best Presentation in Global Symposium in Salt Effect at GSAS 21, organized by the Food and Agriculture Organization of the United Nations on dated 20 to 22 October 2021. He has published one book, Environmental Communication Lab to Land, and Dr. Upadhyay received dozen of students for the MSc dissertation research work, including supervised student. Dr. Sudhir Kumar Upadhyay actively supervise. Sir, we are happy to hear you vast experiments on the topic of salinity, stress management for growth of crop by native salt-tolerant plant growth, promoting rhizobacter, a eco-friendly, sustainable approach. Move to Dr. Upadhyay, sir. Please, sir, start.
Thank you, Shweta. Thank you so much. Sir, Sudhir, sir. Sudhir, sir, you can start your presentation, sir. Welcome, sir.
Okay, thank you. Thank you very much to all of you. Good afternoon to all of you. I am very much thankful to the Shweta who introduced me. Really good things. This is really an immense pleasure for me to share my opinion and thought process at this platform. I think our Vice Chancellor is not available here. Okay, I am very much grateful to very visionary, very inspirational and motivational, our Honorable Vice Chancellor, Professor Nirmala Yash Maurya-ji. Your steps connect to milestones growth of the universities, especially Purvanchal University. I salute you, ma'am, from bottom of my heart. And I am also grateful to the convener of the present workshop, Dr. Noor Jahan-ji, Dr. Ram Narayan sir, Principal of the Guru Nanak College, Dr. M.G. Raghurajan-ji, workshop organizer and coordinator, a very efficient personality, Dr. Dali-ji, organizing secretary and a very hard worker, my colleague, Dr. Manoj Pandey-ji. Thank you all of you for giving me opportunity to share my opinion on management of salinity stress mediated sustainable approach. Well, I can show my slides like how can share my slides. Yes, this is the way I can share my entire screen. Please share my slide. Yes. Slides. Yeah, the full screen I want to. No, full screen. Control F5. No, full slide.
सर टॉप कॉर्नर से जहाँ ऊपर टॉप कॉर्नर पे मिनिमाइजेशन के बाजू में जो है उसको क्लिक करिए, उसके बाजू वाला नेक्स्ट वाला सर.
This one?
Yes sir. Once again. Once
Okay,
again.
okay, okay, okay. I'll wait. Okay, I hope you
Yes.
all see my slides. Yeah.
सर वो
Thank.
स्टॉप शेयरिंग वाला जो बॉक्स उसको साइड कर दीजिए, ड्रैग करिए, वो हो जाएगा.
Okay.
नहीं, स्टॉप शेयरिंग, हाँ उसको जस्ट ड्रैग करिए. स्टॉप शेयरिंग मत करिएगा.
Oh,
अभी
मैंने
stop
कर.
किया.
Hello. I hope
नहीं, अभी
my.
योर स्क्रीन है स्टॉप शेयरिंग सर. कैन यू शेयर इट अगेन?
Okay. Share it again. Hide.
हाँ, हाइड कर दीजिए, हाइड कर दीजिए.
No. Okay. Good. Well. Now, welcome in the virtual world. And today's my topic is Salinity stress management for the growth of crops by native salt-tolerant plant growth promoting rhizobacteria, an eco-friendly, sustainable approach. The concepts of the workshop, I just when I received from Dr. Manojji, then I become very happy to know that the terms, the sentence of the conference to key sustainability. So actually my thrust area is salinity stress management. So I try to connect the sustainability to the environment and our topics. Let's start to discuss my first slides. Life science interface, the key to sustainability. I am really very thankful to those fellows who select this topic of the workshop. This is really incredible. But in my opinion, salinity directly not jumped, salinity directly not discussed, salinity itself having in one umbrella, and that meets three different pillars, then become compiled as the terms of the salinity as a whole. If you think how sustainable approach is arises, what are the requirements of sustainability in the research fields, and why sustainable issues are becoming a burning issues in front of me, in front of world, across the scientists. Let's discuss what are the mindsets of the young researcher have to find out the sustainable approach. So, in my opinion, the first, we try to focus our thought process, our research based on the eco-friendly. Then second, economic feasible, and third, sustainable. The last step is the sustainable. Then we attend the means of the sustainable. Well, why we try to focus first eco-friendly? What is eco-friendly concepts? This is a very common terminology, but an environmentalist can define very broadly to know the eco-friendly concepts. Look, our developments, our scientific developments like DDT, or another energy sector development like Chernobyl accidents, even Bhopal gas tragedies. What happened in those development? What outcome we find? What we realize today? During 19th centuries, DDT was discovered, and DDT have very potentials for the insecticides. But nowadays, we are going to be banned. In 1972, United States itself banned the DDT. Why I try to correlate this concept to the eco-friendly? That is a main things. Kindly listen very carefully. One scientist when develop anything, why that things become very horrible for today's, like a plastics. So many of the things. In Russia recently, everybody is known because the Russia is fighting. Uh, a neighbor country of the Russia related one country is Poland. Since last 30 years back, the milk was banned in Poland because uh, so many of teratogenic effect observed in the newborn babies. The researcher and WHO teams find out the reasons behind of that, and he find out the milk itself have the radionuclei. The government of Poland have no idea how comes radionuclei in the milk of the cow, milk of the cattle, which feed to the children's, which feed to the mother. The research find out that the grass was taken by cows, the grass itself had radionuclei, but the pollens have no any activities, and they are in doubt how our country, how grasslands are become filled of the radionuclei. Then one accident was highlighted that's occur in the adjacent country, Ukraine, in Chernobyl. Nuclear blast occur at the times, and the nuclear fallout occur. The radionuclei escapes, carry through the winds and fall down the Poland's land, and the grass are going to accumulate that radionuclei, and that grass are taken by the cattle, and the cattle milk taken by the females, and shows the teratogenic effect. Well, the effect what we observed after 30 years of the research, and then we are going to be banned such type of the activities. So my focused area about the uh, eco-friendly, a scientist, a researcher, especially young researcher, they must develop their own research on the basis of eco-friendly concept. If we try to focus any outcome from the laboratory that based on the eco-friendly, that will not harmful to the ecosystem and maintain sustainable approach. Now the second aspect is the economic feasibility. This is again a very important concept, because any developments from the lab, from the scientific platform, if that development, that technology not reachable at the bottom of the society peoples, then this have no meaning. In my opinion, this is best of the developments of the technology. So both pillar, eco-friendly and economic feasible technology, are very quite remarkable technologies, very quite remarkable aspects, very quite a thought process of the young researchers to build a sustainable approach. Well, okay, this slide shows the United Nations sustainable approach and 17 agenda. What I showing here, this is already available in UN websites. But the journey of this goal, I try to focus in front of you. How this journey, how we are stated, say that the 17 goal are very important. 17 goal are really meaningful to do something for the sustainable approach. I just try to focus the deep and origin of these goals. I try to inform you. One lady whose name is Rachel Carson. She wrote a book, Silent Spring, a very revolutionary book in environmental science. First time she was highlighted the impact of insecticides on the ecosystem, and that book play a key role for the developments of this type of the agenda and issues. And that book especially uh, key discussion points of book of 1972 World Conference, which was held in Stockholm, and that conference also main reasons we are just celebrating the June 5th for the conservation of environment. So the agenda, so the concepts are arises due to that book only, in my view. And one very important name I would like to share, that is Brundtland. She was the Prime Minister of the Norway, and she gives the concepts of the sustainable approach. She gives the concepts of the sustainable approach, and in her reports published as Our Common Future, with the three different aspects. One is the protection of environment, environmental equality. and social benefit. So the both ladies play a very remarkable role to develop such type of agenda. After 1972, Stockholm, so many of the concerns uh, emerged in front of the world, in front of the scientists. In 1987, uh, HFCs like components are uh, getting more attention because the uh, ozone hole was observed by the Jonathan Franklin in Antarctica. Jonathan Franklin and a group in Antarctica studied and find out in 1985 the ozone hole occur. The reason of that ozone hole is what the scientists just find the HFC, hydrochlorofluorocarbons, CFC, chlorofluorocarbons. The components of these agents, chlorine is very dangerous because they can polymerize the ozone molecule up to one lakh of the molecules. So the journey of the discovery of these CFCs, CFC is very useful components. This is widely used in the cleaning agents, even the refrigeration activities also. But in 1987, in Montreal Protocol, the decision was taken by the several scientists. No, we are going to be ban the CFC and try to find out the alternative of the CFC. Dear friends, just try to point out these two statements. What happened in 1987? Ban on CFC first, and the second is then to find out the alternative of the CFC. It means what? We are trying to focus on alternative researches. Alternative researches for what? For the developments of the mankind, development of the sustainability. And alternative research must be based on the eco-friendly and economic physicals. Similar story happened in 1997 in Kyoto Protocol, when the scientists across the globes try to focus how to reduce the CO2. And similar approach having in 1992 in Earth Summit and given an agenda of Agenda 21. So the sustainable approach is simply what we see in this slides. 17 goal is just a form of the Agenda 21. But the Agenda 21, the Kyoto Protocol, the Montreal Protocol, so many protocol, I am not going to discussing here, are not fit, are not able to perform a real manner, to the root manner. So in 2015, again, United Nations members set up the goal for next 15 years, that is having the 17 goals. These are the no poverty, zero hunger, and so on I have mentioned here. And now, across the globes, the researchers who are working in this field, especially life science and physical science, if the mindsets of the researchers based on eco-friendly and economic physical, then certainly he attained the goal of 17 that mentioned in United Nations Development Program, either UNSDG, and by 2030. So these are the 17 important goals what I showed in these slides. Now please, how this goal we can attain? How we transform the Agenda 2030 by handling of the 17 goals. This agenda must be, you can say, the integrated approach of 5P form. And the reduce, recycle, and reuse concept is little bit differ from this concept because we need an integrated approach to save our environment. One thing I would like to share in front of you. The concept of environment and the word environment is entirely different. Today's we see in a very common in a regular routine practices the environment, paryavarana, pradushan. But the real sense of environment is what? What is the real means of the environment? 1990 in India, actually in our act, the definition of environment mentioned, and they covered only the soil, water, and air. But in my opinion, the environment is not only deal soil, water, and air. These are only the matters which can seen by the naked eyes, and we are realize environment is the illustrator. Environmental definitions need to be rectified, need to be revived, and need to be more open and see through the eyes of the Vedas. However, this is the Agenda 2030 and for the Sustainable Development Goal. And these agendas have an integrated approach of the people, planet, prosperity, peace, and partnerships. The role of the people's roles means what? To the people, by the people. These are the concepts of the agenda, the goal of UNSDC. Even the planets, if you are talking about the Earth, of course, Earth, you are living on these planets, but the interaction of the planets, this planet is not itself alone. This planets is also a part of solar systems. The whole planets are going to be governed the existence of planet Earth. So the planet meets a very vast concepts, not only the very concise way, not only a study to roll the climate change. Why I am saying like that? Dear friends, if you are trying to focus one point, the CO2 is the main points for the rising of the temperature of these globes. Suppose that I am just taking one example, and IPCC also gives so many of the data and showing in front of a different platform the temperature is rising, and the risings because of the greenhouse gases. And the main concept of they confer about the CO2 is the main greenhouse gas. Well, recently I find out several research from the Dutch scientist and find out that the CO2 itself having a retention capacity, even every matter have a retention capacity, it's have a threshold limit. The CO2 have whatever the threshold limit that not able to sufficiently explain the rising of the temperature of these planets. Well, the Dutch scientists correlate the earth temperature rising with the sun hot spots, solar hot spots, and Indian philosophy, even astrological concept, if you are seeing very deeply, then you find out that the earth temperature can be regulated from the sun. But we are, because we are in very limited condition, because science have very limited hands, so we just try to focus as the main culprit to CO2 for the responsible of the climate change, you can say, the temperature rising. So the in sense of the planets, my opinion is that the planet itself, to the planet, by the planet. Next one is the prosperity, peace, and partnership. These are the really social and economical issues and ethical issues. So the 5P concept integrated work together, then we can achieve the goal, sustainable goal, and try to try to fulfill the agenda issues by 2030. Thank you. Next, please. Well, so the purpose of earlier slide is showing have some meaningfuls. I'm trying to connect the sustainable goal to my research activities. I'm the person who work on field of the environment and deal with the microbiological concepts, applied microbiological concepts. Recently, we have observed the rapid industrialization, urbanizations, degraded organic chemicals, extensive agricultural activities, waste disposal, and mining activities, so on. This activity is growing on, and of course, the demand is of industrialization, urbanizations, and whatever the development is under streamlined, is going on. Need and greed and so many of the concepts, but I'm not in that going in that way. I'm just focusing in the real scientific fields. So what I conclude with the enlightenment of the 17 goals, our thrust area, our working area is first, second, third, and 12 and 15, which cover in my research task, because I am drilling the soil pollution and trying to get some remedy, how to restore the soil, how to improve the plants growth, if the cropping system is improves, then scarcity of the food can be minimized. These are the thrust area whether I am working, and especially I am talking about salinity stress in our next slides. So, so the overview about the salinity and the salinity problems, salinity stress problems. Salinization and salinity stress is a common word in agricultural field is a very common phenomenon. And Butcher et al. 2016 reported that about 50% of the arable lands contaminated by the salt. This is a very, very horrible situation, because once the salinization is increased, the plant growth is going to be stunt hampered and reduce the productivity, and scarcity, food scarcity has been arises. So, in scientifically, what is saline soil? So many of definitions are available. You can read through different research paper. But in a research field, in a real laboratory condition, how students can find out this is the saline or not saline soils. If the electrical conductivity of any soils having more than 4 decisiements per mole, may be treated as a saline soils. This given by the USDA, United States Development Authority. In saline soil become less fit for the growth of the crop, because more of the nutrients, available nutrients, are bind and form in a complex forms. So the harmful effect of soil salinizations leads to deterioration of the agricultural soil, dampen crop production worldwide, extensive farming the application and synthetic chemicals, urbanization. These are the factors that are responsible for the salinization. One more things I am also share in front of you. So a few of the practices you can say that the salinizations is done by the man-made activities, but the few of activities done by the geogenic also, and at geogenic activities man cannot be controlled, and whatever the man activities like synthetic chemical uses, urbanizations, uncontrolled indiscriminate disposal of the waste can be managed, salinization. So in these views, I try to find out some data about our country, our country with the respect. So the real saline soil figure not available. One more things, because at root level we are not going to find out the real saline soil status. Anyhow, I just received one data from the Central Soil Salinity Research Institute, Colonization, and data shows in 2015. This data shows that about 15 states of the India having saline soil state, and about 4% of the lands are saline soil affected lands. A very huge amount of the lands. We must be save these lands, because land soil is not increased. You know, the formation of soils, nature takes about thousands of the years, and the rapid population growth directly based on the engineered agricultural systems. And at this stage, horizontal soil must be used, must be applied, must be used for the food productions. So the saline soil management is very, very necessary for the developments of the crop and yield to fulfill the requirements of the food of the coming growing populations. Well, so I just try to connect through this slide with the UNSDG's goals. Soil salinities act as a breaker of UNSDGs. How? Soil salinity is the worldwide concern that decrease plant growth performance in agricultural food and contribute to food scarcity. Even I just read one scientist, really I'm not remember, who alone that by 2050 we are facing a food scarcity. And the reason is one of most important reason is the soil salinity. And so UNSDG goal and soil salinity directly correlated. Salt stressor have adverse impact on the plant ionic, osmotic, and oxidative imbalance, as well as numerous physiological functions. Uh, okay, salinization of course impact bad impact, but the plant itself having some coping mechanism against the salinity stress. Even not only salinity stress, against any type of the stress. This is the natural phenomenon. Plants have its a variety of coping strategies to deal with the salt stress, include osmosensing, osmoregulation, ion homeostasis, increase antioxidant synthesis, and so on. In the past several decades, researcher has shown rising salinities in the agricultural field reduces crop production and plant development dramatically. Yet, plant have a well-known coping mechanism that protects them from salt stress. Well, plant have a well-known coping mechanism. But why production is going to be reduced? What are the basic reasons? If the plants, if the cropping system, if the crop have itself own mechanism to fight the battle on saline field, then why yield is going to be reduced? This is the question for the scientist. This is the questions for the agricultural scientist. This is question for the environmentalist also. Well, this figure I just show. This is my figure I just published in 2022. And this figure is very important for those students who are working in these fields and try to find out the main reasons of the plant stress and damping of the yields of the plants, growth of the plants under the stress conditions. I concluded 7 different aspects directly impacted on the plant under the salinity stress in spite of osmotic and ionic stress. So first is the wilting and chlorosis. Second is altered stomatal activities. Third, altered electron transport system. Next, inhibition of enzyme activities, nutrient imbalance, increased reactive oxygen species, altered electron transport system, and finally, damage the DNA. So the stresses, external stresses play a very negatively, very adverse impact of on the plants. Well, two concepts are going to be correlate here. First, the problem is the salinity. Second, plant itself have fighting ability against the salinity. Third, the outcome of both of the battle relationship, the reductions of the yield, the growth of the yields, then what we can do, what a scientist can do, how we improve the yields of the crop, how we improve the growth of the productivity, growth of the plant performance at that type of the conditions. And the condition must be eco-friendly because we attain the goal of the sustainability. I earlier slide shows eco-friendly, economic, physical. So on that way, the eco-friendly management strategies are become very, very important, very, very remarkable, very, very sustainable. So in our research activities, 2 different aspects I have taken. I have think to manage the problem of salinity in agricultural field. First one is the plant growth promoting rhizobacteria. Even so many of the microbes have ability to plant growth promoting activities, but I am checking plant growth promoting rhizobacteria. And second, eco-friendly amendment. On eco-friendly amendment, I just recently worked since last 3 and 4 years back. But plant growth promoting rhizobacteria have been work since 2007, 6 and 7. And the main feature of the plant growth promoting rhizobacteria are plant growth promoting rhizobacteria are the soil bacteria. They can colonize root. They can associated with the rhizospheric regions. They have capability to promote plant growth by the direct and indirect mechanism. And also the application of these PGPR can decrease the global dependencies on hazardous agriculture chemicals like pesticide, insecticide, whatever. Stimulate plant growth through mobilizing nutrients in soils. Well, dear friends, these are the beauty of the plant growth promoting rhizobacteria. When I started the work with my guides, Professor D.P. Singh and Dr. Anil Saxena, in a very reputed laboratory in NBIM, and the concept what I using plant growth promoting rhizobacteria. At that time, I realized that I have a very good uh, research because we have a solution of the salinity problems. But after the getting some experience, I find out that the solution of the PGPR is not enough to manage the salinity problem at root level. So another hypothesis must be arises. The actual research concept I am focusing in my next slides. Why modifications require in selection of the plant growth promoting rhizobacteria. After overall the available technology, available biofertilizer agents available nowadays in different countries, even in India, based on the plant growth promoting activities, but they are failed to at the field level. Either they are not able to performance. What are the reason behind that? So now we are showing the next slides. The real concept, real hypothesis, how we select the plant growth promoting rhizobacteria. Because since 1980s, the PGPR inoculum have been developed, but the few of them revealed irregular performance at field level. Although several researchers have developed the consortia of the plant growth promoting rhizobacteria, but with more or less similar outcome at formal level. This is very important finding. The solution of this problem is somehow hidden in the root excuded and root microbe environments. Again, the very important concept has been emerged in front of you. The microenvironment, the root environment. Not only a very microbiological concepts, PGPR act in a similar fashion to the all of the plant at different level of the salinity. So these particular concepts arises given and open our eyes to reach in a new horizons. And the next concept I have been using in our studies, eco-friendly awareness. I will discuss later on. Well, dear friends, what I am saying in earlier slides, the PGPR itself not at a very good performance at the field level. Why? Because the selection of the PGPR from a different place cannot act at different place, fails in result at field level. What are the reasons behind that? This reasons is hidden in plant itself. We are not perfectly recruit the PGPR. You know, the recruitment is very important concepts in university system, either in everywhere. If the recruitments are bad, it means what happened in very well. In a frontier in microbiology, recently I have published one paper, root excluded. If anyone interested, they can see, either I can send. The recruited PGPR play a very important role, play a very selective role, play a very effective role at a field level. This has been given one hypothesis after the study of the several research papers. What I saw in this figure, if you see in left side, pointer is not here. If you see left sides, delta PZ, the change in the plant growth stage. The change is the natural phenomena of the nature. Everybody is going to be changed. No one can stay in a stable positions. Every molecule exists in a dynamic positions. The change is the principle of the nature. Similarly, the plant itself having a change in nature. The different stage of the plant is different stage of the plant itself have a variable and variable with the respects of the change of the environment. You know, the environmental change is the basic principle of the nature. Change is the basic principle of the nature. But recently we are more focusing about the changing of the environment. What the difference between the change and the changing of recent change in environment? The change is the natural principle, yes, but the recent change is the meaningful, because the dynamics of the change of the environment is going to be triggered, and that is the main problem. If you see the principle of the developments of the phylogenetic development, environment plus gene produces phylogenetic. This is the true. This principle is the basic principle of the developments of the evolution. But nowadays, rapid change of environment is a big problem. So, what I saw in this figure, delta PG and delta EF, change in the environmental factors and change in the plant growth stresses. Rapid change of environmental factors create any stress, and once plant is going to be stressed, either in general, the plant have itself own tendency to do the photosynthetic, and 25%, 5 to 21% of the photosynthetic matter released through the root tip, and that photosynthetic matter itself have a very complex nature of the organic compounds, very complex nature of the signal molecule, generally we called root exudates, a very complex, a cluster of the compounds. But that compounds have a capability to select the particular plant growth promoting rhizobacteria. This is the important phenomena, what I am saying, what I am trying to share in front of you. Any PGPR, any plant growth promoting rhizobacteria, why failure at different place? That is the basic reason, because the selection of that particular plant, we are not going to make any minds. If we are going to follow the process of the selection of the root exudates compounds to the PGPR, certainly we get the better result at field conditions. The failure of the field conditions is the lacking of these concepts. So this figure shows what? The photosynthetic matters having the different type of the compounds. That compounds have a osmosensing, osmoregulations activities. That compounds also itself having the nutrients. That compounds having the ability to sense the the plant growth promoting rhizobacteria with the classical mechanism of the Curum sensing, and then select a particular PGPR, particular plant growth promoting rhizobacteria, like any bacteria like Bacillus, Azotobacter, Aspergillus, whatever. No, Azoospermum, not Aspergillus, and they colonize. So the root exudate have a ability to colonize and select the particular strands. If the researcher work through, if the researcher find through the selection of PGPR through this process, their chances of failure at field level is going to be minimized, greatly minimized. Once PGPR colonize, adheres, I earlier shown in slide, but the nature of the PGPR is the root colonization. Once the PGPR, plant growth promoting rhizobacteria, colonize on the root hairs, they active, they develops, and they works direct and indirect mechanism to plant growth promotion. Direct mechanism is like phytohormone productions, like indolestic acid, gibberellic acid, so many of the enzymatic activities, ACC deaminase activities, nitrogen fixation activities, phosphate solubilization, potassium solubilization, even different mineral solubilization activities also find in these bacteria. And indirect mechanisms like antibiotic production, siderophore, acetylene production, ammonia, hydrolytic enzyme production, exopolysaccharide production, ISR induced systematic resistance, etc. So, if the workers of these fields try to find out that selection of PGPR through this process, this is selection of PGPR called native PGPR. In my previous slides, in my first slides, if you see, I use the term native, N-A-T-I-V. Native means the particular organism survived at that particular place without showing any adverse conditions. So the selection of native PGPR through root exudates is the perfect way to find out the developments to boosting the biofertilizer technology at a root levels. Next slides. Again, this figure have been published recently. This figure shows that a direct mechanism of the plant growth promoting rhizobacteria. No, any mechanism not initiated itself. A few of the factors are responsible. I earlier tell you in previous slides, the environment is the external factor that have ability to triggers to function the cell, to triggers the function of the life, to triggers the function of states, to triggers the function of any molecules. Because environment is the surrounding of whatever you have to take in. Even surrounding of thought is also environment. So if you see the top of this figure, EF, environmental factor like drought and low nitrogen content condition, I am taking as example to show in this slides. One is the drought conditions and second is the low nitrogen conditions. How a PGPR act in drought conditions, plant sense the drought conditions like in Arabidopsis, and particular bacteria Chlorophyll is going to be attracted and perform different activities, and that activities belongs to direct mechanism like IAA productions, phytostimulation, biofertilizer activities, and so many of the gibberellin compounds. So these mechanisms done, of course, done by the PGPR, but triggered by the condition, and that conditions of come from the environment. So the environment is the key player. Sorry. External factor play a very key roles to manage the activities of the plant growth promotion activities. In the right hand side, low nitrogen content. I am just showing here at low level of the nitrogen content, the flavonoids is releases. Flavonoids is released from the root exudate. Root exudates have the different type of the flavonoids, and that flavonoids recruit rhizobia. You know, any rhizobacteria, any rhizobium cannot act at any place of any pulses croppers. If you are trying to work, then of course failure is you just observe the failure rates. But when you recruit the rhizobia, it means at low nitrogen conditions, root itself release flavonoids and special flavonoids, like so many of the type of the flavonoids. If that particular flavonoid recruit that particular rhizobia, and if that rhizobia you are using in that particular plant, then you can say that yes, that is the native isolates that may improve the plant growth, that may act as a direct mechanism, either the indirect mechanism. So the selection process is very important of that particular rhizobacteria, because if you are not doing in this way, it means we are fell on the compatible issue, and the compatibility issue is a big issue for the organism. Some scientists also call this type of the interaction crosstalk interactions between the root and microbes. So these organisms, this slide shows the direct mechanism of the organisms, PGPR. Well, another slides I am showing here the indirect mechanism. The similar functions again triggered by the external factors, like I am showing here one root is suffer from one pathogen like Rhizoctenia solani, and Rhizoctenia solani triggers different type of the microbes like Pseudomonas fluorescens, and that Pseudomonas fluorescens releases pyrroerythrin type of the components, and that kills the pathogens Rhizoctenia solani. These are again an indirect mechanism of the plant growth promoting microbes. Similar happening in the case of the sidro-4 compounds, defense related proteins, and so other activities that fall in indirect mechanisms. Indirect mechanism also promotes the plant growth. But direct mechanisms impacted directly, and indirect mechanism partially impacted on the growth of the plants. Well, so the earlier slides shows the dimensions of the sustainability, develop the mindsets of the sustainable concepts, how a researcher works, and I try to correlate scientist problems to the uh, sustainable goals with the microbes, how we attain. And now our slide shows how real research formulation requires, how we achieve the real goals to mitigate the salinity problems. Our next slide is shows the formulation of research activities, especially for helpful for those who working in these fields. So objective is very important concepts of any researches, and the previous slides just only gives you informative how to develop the hypothesis to do the work. So in a present research work, I just shown few of the objectives. First one is the isolation of bacteria. Second one is the plant growth promoting attributes. Third one is the evaluation and selection of salt tolerant plant growth promoting rhizobacteria. And one more thing is I would like to share in front of you. In microbiology, essentially, without characterization, without molecular characterizations, not significance you can achieve. So the molecular characterization is must nowadays. So I am just showing few photographs, few techniques how to molecular characterizations can occur. And lastly, the evaluation of salt tolerant plant growth promoting rhizobacteria, how mitigate the wheat plants stress and improve their growth. So these are the objective of the present research works. Now the first okay. Well, so the survey and sample collection. In my previous study, I just saw the few places of the Uttar Pradesh whether I have collected few samples of the rhizosporic soil. One more important thing is that sample collections. Sample collections for getting either the generating a discrete data is very important concepts. You are not only just to go in field and take a sample and just you say that the sample collections is fixed. No. The concept of sample collections must be based on your hypothesis. What type of soil sample you required, why you required, it is really a very matter of the discussions. When I work on, suppose that I am working on wheat, it means on wheat and try to mitigate the salinity stress of the wheat. So we find out the soil sample of a saline infested zone of whether the wheat is cultivated, and find out those plants who have ability to grow in saline soil, saline field, saline infested zone. So I collect the root of the particular plant in those area and collect the soil sample in plastic bags, polythene bag, and then proceed further. So the collection is very important. In saline soil field, this is again a very important concept. You must find out the particular plant and particular rhizospheric soil sample. Otherwise, what happened? You didn't find a real bacterial who are working and might be failure in further researches. So in our research activities, I have introduced different type of the media for the isolation of plant growth promoting rhizobacteria, and different type of the media is required because of the diversity of the microbes. I try to get more. So the nutrient and Kingsby Jensen increase soil extract, soya agar we have using, and we find out that 42 isolates that we are able to survive at 8% of NaCl salt tolerance. And I just target on that particular rhizobacteria, 42 isolate. I just left 32. I just use 42, that is 8%. I'm just taking the higher concentration of the percentage. Well, this slide shows how we screen the salt tolerant bacteria. These are the spot test. This is the very common test can done in laboratories, and this is based on the different concentration of the plates, whether the 20 microliter of each inoculum are going to be added of the bacterial strain, and find out that the survivability of that particular bacteria at different concentration of salt. Well, these are the slides that shows phosphate solubilization activities, hydrophore activities, amylase production activities, cytochrome, and these are the different activities. Few of them I am not showing here. These are the plant growth promoting attributes and osmolytes activities of the salt tolerant bacteria. Well, this is again a very important parameters. The growth kinetics. Suppose that one bacterial isolate show that 12%, 10%, 8% of the survivability for 20 days, for 10 days, for 2 days, for 2 hours. What happened? What kind of the isolate we have taken? So it is must be know that the survivability of the microbes against the stress must be screened. For any researcher, it is recommended. And not only recommended, nowadays the people are working in different concepts like using the friendly Timken and Leninger and pseudo orders reactions. Then any general accept your papers. But at that time I am using only the growth kinetics for knowing the generation time of the microbes at different salt concentration, and find out that only 24 rhizobacterial isolates have a capability to survive higher concentration that may be used at a field level. Now, so maybe used at the field level for what kind of the plants is required. For that we have selected a different kind of the wheat varieties, different wheat cultivar varieties, and screen for their salt tolerance. Dear friends, one point you must be noticed that agricultural scientists, especially the plant breeders, even the uh, GMOs type developments, even the recently developed technology, CRISPR-Cas9 technologies, are working on the improvements of the different breeds of the seeds, different cultivars of the seeds who survive under the stress conditions. Although these are different section of the, this is not my discussion sections, but I have chosen at the times to find out the real result of our isolates. I have screened few different varieties of the wheat cultivar that found in Uttar Pradesh, and find out that the K7903 cultivar variety of wheat survive at a high salt concentration range, and I have chosen this stage for our plant microbe interaction studies. Well, this is the pot experiments, this done in NBIM, and this is a very important concepts of the work to test the inocula, uh, particular plant growth promotion activities by the using of particular strains, because these conditions must be maintained aseptic conditions when you are going to inoculate it that particular strains. So this stage is a very important crucial stage, because this stage is the bridge between the field and lab condition, and I find out some of the important results in that particular 21 strains, studied strains. And these are the colony morphology which I uses in our studies, and further I sequence of this bacterial strains and submitted in gene bank. Well, and I earlier tell in previous slides, without molecular characterizations, the microbes cannot get recognition nowadays. You must be have accession number, you must be have sequenced molecular characterized concepts. So the steps of the molecular characterization, you have to isolate genomic DNA, and then you have to amplify the conserved gene. In bacteria, the gene generally, the 16S rDNA is the conserved gene people are using, widely using for the getting the diversity analysis and getting the identification as a kit, as a tools. And in our study, I have also used this type of the 16S rDNA gene amplifications. One more things in plenty of the isolates, all isolates are unable, thankless job to identify. So you must be develop another approach like clustering. You try to make a cluster of that particular microbes on the basis of RFLP, restriction fragment length polymorphism techniques. While I have using the tetracutters of the restriction enzymes, ALU1, MSP, and A3, and this particular crops of RFLP give one a dendrogram and give some clusters, and these clusters also help to select the particular strain to sequence, and sequencing is the very expensive concept, very needs more money. So there is no needs to sequence all of the bacterial isolates. That's why the RFLP play a very important role to uh, give the clusters, and each cluster, if you are taking on strains that may be present your bacterial of the neighbor cluster zones. So the cluster analysis is important aspects before doing the sequencing. This is also an economic feasible approach, scientific approach. Well, the concepts and the published uh, gene published sequence in gene bank I have published in the very reputed journals, the current microbiology. If you see that particular strands have their accession numbers, these are the Bacillus pumilus, Bacillus equimoris, Bacillus pumilus, and so on, Bacillus spermothermodurans, and so on. Well, now the next steps. The two step is what? One is the isolation, second is the salt tolerant activities screenings, and PGPR activities screening, pot experiment screening, and now the phase to reach at the field level. You know, field condition and the experimental field condition is entirely different from the normal field condition which you see in the farm and daily routine life. So you must be design a field condition very scientifically. For that, random block design is very important. So in our experimental condition, I have found few of the bacterial strains and formulated different type of the RBD design methods. And this experiment was carried out at NBIM because whether the facility I have acquired. These are the single culture inoculum strains, and I find out the isolated. You have to see the different type of the growth. These are fields are 3 by 4 meter square in range, prepared by the folios, and I have using our culture at that particular field. These are the field conditions, and I have find out that few of the good result by using of the single strains, and you see here after 60 and 90 days of total dry weight differences, and find out that the three isolates, especially 1 to 1 isolates and isolated 24, isolate 44, are very good. But and this is the granule of using the single culture inoculum experiments. Bacillus subtilis, Bacillus pumilus produces more granule under the saline conditions, more growth of the wheat conditions. But the story is not end. This is the experiment of one culture experiment. That's maybe failure at the field level. That's maybe worked at the field level. Doubtful condition arises always. So the scientists, the researchers, the hypothesis change the patterns to develop a single from the consortia concepts. And now again we develop the consortia on the basis of the compatible behavior of the microbes. For that I have using the compatibility test between the selected isolate and find out different compatible conditions and prepare different combination And here you can see the treatment what I mentioned here for the different combination. These combinations are based on the agar plate, Na media plates, different by using of the different culture and the computability behavior at lab. And finally, we find out that the Bacillus pumilus and the Bacillus cecomerus and the Bacillus subtilis, these are the all Bacillus group of the strains in treatment T9s produce more yield, more growth. And similarly, in treatment T3, whether the isolate 1, 2, 1, 8, and 47, Bacillus pumilus, Bacillus mendocina, and Arthobacter species again produce the more yield, more grain yield of the wheat, chosen variety of it under the salt condition. So the stories again shifted toward the single cultures to the consortium developments. Well, dear friends, the mechanism behind of these stories I have published in the plant biology, and these are the simple figures, and this figure states that the concepts of the papers, how particular microbes helps. If you see in this figure, here left side the saline soil, Si soil initial sodium, or Pi plant initial sodium in it. This shows the adverse condition. But by the adding of the STPGPR, the sodium of the saline soil shifted in STPGPR, either the bind through STPGPR, and the pressure of the wheat plant, sodium pressure of the wheat plant are reduced, and plant growth are going to be increased. Not only the plant growth, dear friends, I am thinking about not only the plant growth. We are talking about the sustainable approach. Our soil health is also necessary. Soil health is also important factors. So the plant growth index and soil health index is also improved by the using of the salt tolerant plant growth promoting rhizobacteria. Well, dear friends, recently we are working. I'm just showing in our first slides. I'm working on PGPR as well as eco-friendly amendments, and recently, since last 2 years back, I am working on PGPR. Simultaneously, I am using some eco-friendly amendment like poultry manure, spent mushroom substrate, and cow dung. And I find out that the poultry manure and spent mushroom substrate have a capability to grown, have a capability to बात कर लीजिए. Hello. Hello. हाँ. Hello. 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 बात कर लीजिए. Hello. Hello. What happened? Hello.
Yes, sir, you are audible.
Audible. Okay. Only 5 minutes I will take to finish my lecture, please. Okay. Okay. So recently I am just working on PGPR by using some eco-friendly amendments like poultry manure, spent mushroom substrates, and cow dung. And I find out that poultry manure and spent mushroom substrate have a good capability to growth the salt tolerant plant growth promoting rhizobacteria, and that have ability to promote plant growth. Especially I'm working on maize, and this work published in the very good journal of environmental researches. And these are the data which is published recently in 2022 by my students, PhD students. And if you see left side, one graph I am showing, this graph made by the ArcGIS on the basis of secondary data as well as primary data. The condition of Jaunpur is not good enough. The soil salinity, the saline soil status maybe play a very critical and danger role in near future about the Jaunpur. I have found in our studies and presented and published in the research papers. Well, dear friends, the real outcome, what I conclude in only few words, few sentences. The hypothesis is of specific recruitment would address the key gap of the wanting the perfect plant growth promoting rhizobacteria candidate and opening new horizon of the research in biofertilizer technologies. It would be a promising technique for reducing the asymmetrical performance of the plant growth promoting rhizobacteria at formal fields. So I hope you will be all getting little beneficial. Thank you very much for the patience hearing. Thank you. Thank you very much.
Thank you, sir. That was indeed a very explanatory thing on your topic. It was. I think the experts would be better to tell what it was. We will continue to the next. Thank you, sir. Thank you so much. We will continue to our next session. I again invite Ms. Shweta Singh to introduce the resource person. Shweta.
Thank you, ma'am. Thanks again. So going to introduce Dr. V. Lavanya as a resource person of second session of 5-day workshop. Dr. V. Lavanya working as an assistant professor at Guru Nanak College, Velachery, Chennai since 2020. Dr. V. Lavanya completed her PhD in biotechnology. She got the position of SRF in the Department of Animal Biotechnology, Tamil Nadu, Veterinary and a Science University, Tenuvas, Vepri, Chennai. She has technical specialization in molecular techniques, animal cell culture, and protein purification. She has published 15 research papers in the international journals and also have a 11 Scopus Index. I request you, ma'am, please share your experience on the topic plant and animal biofarming role in sustainability. Please, ma'am.
Thank you, Shweta. Lavanya, ma'am.
Yes, Dolly, ma'am. Thank you, Shweta Singh, for that introduction. I hope my slides are visible.
Yes, ma'am. Visible, ma'am.
Yeah. Thank you for the confirmation. So, a very warm evening to one and all. Yeah, a very warm evening to one and all. And I would like to thank all the organizers for giving me this opportunity to share my opinion on the topic life science interface, the key to sustainability. And also huge thanks to all the participants for joining in. So my specific topic for today's discussion will be plant and animal biofarming and the role in sustainability. Okay, to start with, environmental degradation, which is caused mainly due to deforestation, industrialization, urbanization, and the ever-growing population are the two main problems on the planet Earth. So with increasing population, there is also an ever-increasing demand, not just for food and water, but also for drugs and vaccine. And it was very clear even during this corona pandemic. So fulfilling the demands of growing population is actually difficult from the limited land which is available on the globe. Yes, it is of utmost importance to cater to the needs of the present generation. But what about sustainability? So biotechnology is actually known to contribute to sustainability, both by reducing the land area needed for agriculture and also by making nutrient use more efficient. So what exactly is sustainability? So the basic definition is meeting the needs of the present without actually compromising on the ability of the future generations to meet their needs. So to make it more simple, we actually need to be very conscious that we do not deplete the natural resources which are actually meant for the future generations. So though we may have an increasing demand, we should be like self-conscious. All the individuals on the globe need to have that awareness. So biotechnology provides the building blocks for a sustainable future. For instance, the drug development process within the pharmaceutical industry has actually undergone a very significant transformation over the last few decades. And this is largely due to all the advances in biotechnology. Genetic engineering, which is considered as one of the vital tools of biotechnology, has been used to modify plants, animals, and microorganisms according to our desired needs. So genetic engineering actually facilitates the transfer of desired characteristics into other plants and animals. So coming to my topic, biofarming is one important way by which genetic engineering helps us to cater to our increasing demand, but in a very sustainable fashion. So what exactly is biofarming? So biofarming actually refers to the use of genetically engineered animals or plants as biofactories to produce pharmaceutical proteins. So pharmaceutical proteins can be drugs, vaccines, antibodies, antigens, enzymes, hormones, etc. So this actually, the entire process of biofarming involves the insertion of the specific gene which codes for that particular therapeutic protein into the plant or the animal cell. And here, as the products are derived from the living organisms, they belong to a class of pharmaceuticals specifically known as biopharmaceuticals, bio meaning life. The genetically engineered plants and animals are actually considered nature's cheapest bioreactor, or in more simpler terms, cheapest factory for producing the recombinant proteins. So why exactly do we need biofarming, and what exactly are the advantages associated with it? So biofarming facilities actually help us to overcome the problems associated with the large existing bioreactor systems. So these can actually replace these expensive bioreactors which are actually used by the pharmaceutical companies to produce the pharmaceutical proteins. So one, they are very large, and moreover, they are very expensive as well. So bioreactors can actually be used to produce the expensive pharmaceutical molecules by just expressing them in plants or animals through the insertion of the specific gene which codes for the protein. It also provides a cheaper alternative of extracting the desired molecule. So bioreactors basically help in production of the protein. So once they are produced, they have to be further processed, like they have to be extracted, purified, and further processing is required. So biofarming also provides a very cheaper alternative of all these process, and most importantly, in a very quick way. So the entire processing and purification is very rapid. So what exactly happens is this results in a very quick production of the pharmaceutical protein on an industrial scale. So thus it is possible for us to fulfill any emergency demand, if any. So these are like a few key milestones of biofarming. So way back in 1990, the first plant-derived pharmaceutical protein, the human serum albumin, was produced using transgenic tobacco and transgenic potato plants. In 2006, the injectable plant-made vaccine for Newcastle disease, which is known to affect the poultry, was made using tobacco plants. So this vaccine against the Newcastle disease virus was also approved for use in the United States. In 2012, LISO, the first plant cell-based enzyme replacement therapy, was approved by the FDA. So the concept is LISO is based on the use of transgenic carrots to produce a recombinant enzyme, and this recombinant enzyme is actually used as a replacement therapy in people suffering from Gaucher's disease. And most importantly, in 2014, ZMapp, the drug which was produced in transgenic plants, was successfully used to treat human patients who were infected with the Ebola virus. So coming specifically to plant biofarming, so plant biofarming refers to the use of genetically modified plants alone to produce a wide range of pharmaceuticals. And here the products made will be specifically termed as plant-made pharmaceuticals. So this plant biofarming actually helps to overcome the problems which are associated with the existing bioreactor systems. So at present, the two commonly used bioreactor systems are the mammalian-based systems and the bacterial expression systems. So yes, mammalian-based systems, they have got their own advantages, like they are very efficient. So the recombinant protein or the therapeutic protein which is produced can actually be produced in a very efficient way. It is like it comes in a fully processed way. But the drawback associated with that is that they are very expensive, and scaling up of the protein, like mass production of the protein, is also not that easy. So coming to the bacterial expression systems, so bacteria, yes, they are known to grow really fast. The doubling time is really quick. Also, they don't need any complicated media. So they can actually grow in very simple media as well. So yes, scaling up of the protein or the mass production of protein is easy. But again, in bacterial expression systems, the proteins are not completely processed. So they are more in a very raw form. So it is up to us to actually isolate, extract the protein from the expression system and then go for further processing. So plant-based system or plant biofarming helped in overcoming all these drawbacks. For example, plant systems enables efficient processing of the protein and also allows large amounts of proteins to be purified at the industrial level. Plant biofarming is definitely not new to us, and medicines from plants are also known to be used since many centuries. So the plant as such has got medicinal values, and drugs were also extracted or derived from the plant itself. So earlier, the plant-made pharmaceuticals or plant biofarming, they focused only on using food crops because they felt it is easy to use food crops such as corn and rice, so that they can create a very cheap and efficient way to deliver the vaccine. Just taken the food and the vaccine is delivered orally. But later, tobacco and other non-food crops were also used to produce vaccines. So they will be literally grown in a very controlled environment, and later the pharmaceutical protein will be obtained from it, purified and given in an injectable form. So this plant-based molecular farming has been exploited during epidemic and pandemic as well. So during the influenza pandemic, influenza-like virus particles were actually produced in transgenic tobacco, and these were most importantly found to elicit very good immune response. So they were found to be very effective as well. And later, vaccines against human immune deficiency virus, Zika virus, and Ebola virus, we have already seen ZMAP, were also developed. So little about insulin, which is actually the first human recombinant therapeutic protein. Insulin, yes, it is a hormone which is produced by the beta cells of the pancreas. So I think most of us, all of us know at least another person who is suffering from diabetes. So in diabetes, the particular person will not be able to secrete enough insulin. So insulin actually has to be given from an external source so as to keep their sugar level under check. So earlier, insulin was actually extracted directly from the pancreas of killed cattle and pigs. So the cattle and pigs were reared only for this purpose. They were sacrificed, and finally the insulin was extracted from the pancreas. So just from the sound of it, we know there will be many ethical concerns associated with this. Apart from that, there were also many other shortcomings. For example, the insulin which was directly extracted were known to stimulate allergic response in certain individuals. Again, biotechnology actually proved to be the savior, wherein recombinant DNA technology enabled mass production of insulin. So this is a pictorial representation of the production of insulin by the traditional method. So here, very briefly, the gene coding for the insulin protein is obtained from the pancreatic cell, and then we will take a plasmid from the bacterial cell. A plasmid is basically a vehicle which is going to carry our gene of interest into the host cells. So both of these will be combined, resulting in a recombinant DNA or a hybrid DNA. So this hybrid DNA with the insulin gene is then inserted into the bacterium. The bacterium, since it has got a foreign gene now, it is referred to as a recombinant bacterium. So the recombinant bacteria are then allowed to multiply in a fermentation tank. So as they have the gene coding for insulin, they will be producing the human insulin. So insulin can then be extracted, purified, bottled, and now it is ready to be used. But again, recombinant human insulin was mainly produced either in E. coli or the yeast Saccharomyces cerevisiae for simple reasons that they are very simple organisms which multiply rapidly. But current method of this production of insulin required vast investments. Also, again, there is an increasing demand for insulin. So the demand for insulin is only going to go up exponentially. So there is actually a compelling need for more efficient methodologies. So here again, recombinant human insulin has been successfully expressed in oil seeds of Arabidopsis thaliana. So here the insulin will be produced in the seeds. So one more advantage is the seeds actually also help to store the insulin. So they don't need to be specifically stored anywhere. So it can be stored until required, after which it can be taken and further used. So here I have just shown the steps in plant biofarming. So yes, the candidate gene or the gene which is coding for the therapeutic protein will be inserted into the vehicle, after which it will be inserted into the plants. There are various techniques which can be used depending on the plant. So if the plant is an edible one, the vaccine can be orally administered and the person is going to be immunized If not, the plant can be further purified, resulting in injectable vaccines. So topical application of the vaccine was also possible. So again, the concept of using plants to produce recombinant pharmaceutical proteins is not something new. So there are so many proteins. I have just mentioned a few here. So these are a few pharmaceutical proteins and the transgenic plants in which they were produced. So these pharmaceutical proteins are actually in various stages of development. Not all of them have been approved, but a few, like the antibodies against the hepatitis B virus, which was produced using transgenic tobacco, has been approved for use in Cuba. Again, antibodies against Streptococcus mutans, which was produced using transgenic tobacco, was approved for use in Canada. And ZMapp, yes, it has already been approved by the FDA. So these are certain other pharmaceutical proteins, and I have more to come. So these pharmaceutical proteins, these are like more of success stories. They were found to be very effective. So looking at all this, the researchers were actually curious to exploit the potential of plant biofarming to help design and develop effective vaccines, therapeutics, or drugs to counter COVID-19 as well. And yes, right now we have a vaccine, Covifense, which is the world's first plant-derived COVID-19 vaccine, which was quite recently cleared for use in Canada. So right now it can be used only in Canada. And most importantly, it is found to be like 70% effective against 5 COVID-19 COVID variants. So this is a pictorial representation on the production of the plant-based vaccines to combat COVID-19. So like in the other methods, the gene which codes for the spike protein is allowed to be stably or transiently expressed. So here we can see like there are different methods by which the gene can actually be inserted into the plant cells. So we can actually go for physical methods such as electroporation or using gene gun. So gene gun using which the gene is directly bombarded into the plant cells. So we can also go for certain plant virus which will act as an intermediate to transfer the gene into the plant cells, and the traditional way of using a plasmid. So the transgenic plants with the antigenic gene will then be produced, and again depending on whether it is an edible plant, the edible part with the vaccine can be orally consumed, after which the person will be immunized, or the vaccine can be purified from the plant, resulting in injectable vaccines. So these are the advantages of plant biofarming. So plant biofarming actually serves as a cost-effective production system of recombinant proteins. So compared to the traditional methods, they are definitely cost-effective, and they can be used for long-term storing of vaccines as well. So like we saw in the case of the seeds, they can actually be used to store the vaccines. So this in turn will save us the cost of processing or further purification. Now certain plant tissues are actually suitable for oral administration, thus minimizing the cost and the labor which are associated with the delivery of injectable vaccines. And plants like banana, potato, spinach, etc. have already been used to produce vaccines against cholera, measles, hepatitis B, etc. And most importantly, these edible plant vaccines are known to be comparatively safer than the traditional counterparts, because in the traditional way, we either use live attenuated or the killed virus as the vaccine. Okay, coming to animal biofarming. So again, this is like the farming of animals that have been genetically modified to produce the pharmaceutical substance. Again, here the substance will be referred to as a biopharmaceutical. So the animals in which the genome has been altered, they will be referred to as transgenic animals. So here it can include cow, sheep, pig, goats, etc., which have been modified to produce a substance in their milk, blood, seminal fluid, plasma, urine, etc. And it can also include chickens, which have been modified to produce a substance in their eggs. So these are the steps which are involved in animal biofarming. So to start with, a hybrid gene has to be created. So over here, the goat regulatory sequence and the human antithrombin coding sequence, which is actually the gene of interest for which we need the protein, they are joined together, and here specific enzymes known as ligases will be used. Ligases are enzymes which will help in joining together two genes. So the recombinant gene or the hybrid gene so produced will then be injected directly into the goat embryos. So here in the picture, the technique which is shown is microinjection, wherein the hybrid gene is directly injected into the goat embryos using very sharp needles. This is in fact the technique which is also used in human in vitro fertilization. After this, the goat embryos will then be implanted into surrogate mothers, and once the embryos are transferred into the surrogate mothers, they will give birth to the transgenic offspring. So the recombinant antithrombin gene will be expressed only in the mammary tissue. So this is very important. So we have certain promoters which will make the protein to be expressed only in the mammary tissues of the transgenic offspring. The protein will be secreted only in the milk and not in any other fluids, thus making it really safe for the animals as well. So antithrombin protein, which is expressed in the milk of the transgenic females, can then be isolated and purified, further purified. It can be used in treating antithrombin deficient people. So these are the advantages of animal biofarming. So it is like much safer than actually extracting the proteins directly from the living or recently dead organisms. And the scalability is also really rapid. So if we have to go for mass production, it can be done at a very quick rate. So the manufacturing facility cost is really less as compared to the traditional methods. And the safety is also high because there is very less risk of contamination with cross-contamination with animal or other human pathogens. And it has also got the ability to produce novel and complex molecules. So we can actually play along with it. The gene can actually be altered according to our need. And the recombinant proteins are isolated from the various production systems of the transgenic animals, including the urine, milk, egg, plasma, etc. So though there are so many other production systems, milk is actually considered to be the most preferred source of production system. The reason being, milk is like easily collected in very large volumes. So it can be easily produced and further purified. Moreover, any foreign protein which is made to be expressed in milk is done in a very huge quantity. So expression of protein itself is very efficient. Thus, scalability is very easy. If you have to go for mass production, it becomes really easy. The most important point is the absence of any active proteases in milk. So proteases are a group of enzymes which will degrade the proteins. So we should not forget that our protein of interest should not be cut by any enzymes. So they should be stable. So here the absence of any protease sees to it that the protein of interest is stable. Animal biofarming, yes, it is already a reality with many drugs already having been approved. So the antithrombin alpha drug, which was approved for treatment of congenital antithrombin deficiency, was produced using transgenic goats. C1 esterase inhibitor, which was approved for treatment of hereditary angioedema, was produced using transgenic rabbits. And Canoma, another drug which was approved for the treatment of lysosomal acid lipase deficiency, was produced in transgenic chickens. So most importantly, antibody cetuximab, which is sold under the commercial name Erbitux, was approved for the treatment of metastatic colorectal cancer and also head and neck cancer. So here metastatic refers to the fact that the cancer will not be confined to any specific organ or tissue. So it is rapidly spreading. So this itself makes this drug more efficient. So it is actually used in the treatment of metastatic cancers. And this particular drug was also produced using transgenic goats. So yes, scientists have already proved the successful multiplication of many pharmaceutical compounds and vaccines, other antibodies, beneficial proteins, etc. by biofarming. The biggest ever challenge solved was the mass production of the drug ZMapp, which was used against the Ebola virus and using transgenic tobacco plants. However, biofarming is not yet received properly. It is so far received little attention among social scientists. And the biggest challenge is the non-acceptance of biofarming and transgenics as such by the government as well as the society. So yes, as the earth approaches its carrying capacity for human activity, I feel we must adopt more sustainable ways to generate, distribute and consume resources. And biotechnology is definitely critical for a very sustainable future. Thank you. Thank you for giving me this opportunity.
Thank you, ma'am. That was a short and a crisp presentation, but very clear. Participants, the floor is open for questions. If you have any questions, please, you can directly interact. The people who are in Google Meet can directly interact with the resource people. And those on YouTube can post your questions in YouTube. We'll pick it up from there. Any questions? Noorjahan ma'am has, ma'am, you're mute, ma'am. Noorjahan ma'am, you're mute.
Can you hear me, ma'am?
Yes, ma'am.
Yeah, I thank both the resource persons. So both the sessions were very useful and informative. I have a question for Lavanya ma'am. Ma'am, after that, whatever you said, no, the biofarming, what they'll do to that animal, madam? Whether it will be, what is the fate of that animal? What they'll do?
Ma'am, they are very careful that they are not left to mate with other animals as such, because we have to be very careful that they are reared separately, and in most cases they will be used only for this purpose. So sacrifice, yes, at one point, but no other use of the animals is possible.
So even for the plants, the
Yes,
seeds
ma'am.
and whatever we are getting whatever other products we won't be using, right?
No, ma'am, no, ma'am. They will be used only for this purpose, because again, cross-hybrid varieties will be harmful, and we need to preserve our wild varieties as well. So they are very careful that they are not allowed to be mixed with the wild varieties. So they are reared separately, and they are very careful that they are not left out.
Yeah. So
Same with the animals.
Yeah, it should be safeguarded by government policies, right?
Yes, ma'am. Yes, ma'am. All the countries should have their own regulations first, and that is very important, because there are a lot of ethical concerns.
Okay, thank you, ma'am. Thank you for the very informative session. Thank you.
Ma'am, there is a question in the chat box here. Vishakha ma'am has asked that when we are talking about biofarming, where we all know the plants process proteins differently than animals or humans, the body might recognize a human protein produced in plants as foreign, allergic reaction, and create situation of epidemic. You can see the chat box, ma'am. I think it will be
Yes, ma'am. Yes, ma'am. I'm going through the question. Yes, in such cases, during clinical trials or pre-clinical trials, if it is found to cause any allergic reactions, we need to definitely go for further processing. Because when we use animals, it's like more related to humans. But using plants, yes, this problem is there. So if it is not completely processed, we have to do some post-processing work also, like before the actual purification process. And before it comes to the market, there are a lot of preclinical trials, clinical trials. So yes, and that is one of the reasons why we actually do not have a lot of products, plant-based or animal-based products in the market. So initially it might be showing good results, but after that, this might be one of the reasons.
Thank you, ma'am.
Madam, any biofarming application in fishery sector?
In fisheries, yes, sir, there are some very preliminary work going on. So it
Okay,
should be. Yes, sir. Yes, sir.
Okay, thank you, madam.
Ma'am, there's another question in the
Yes.
chat box, ma'am.
Yes, ma'am. Yes, sir. It definitely has a lot of scope for budding biotechnologists, and I feel like biotechnologists should actually come out with how to overcome the possible risks of biofarming as well. So I would not say there are no risks concerned at all, but young people should come out with ways to counter those risks as well. And it is definitely has a lot of scope for young people. And this has been around since the 1980s. Then we must start working on this, sir. Thank you.
Thank you, ma'am. Any other questions? Okay. So this brings us to the end of today's sessions. I thank Dr. Sudhir Upadhyay ji and Dr. Lavanya ji for their time and the wonderful presentations they have given relating to our workshop theme. We will catch up again tomorrow for day 3, where we'll have Dr. S.P. Tiwari sir and Dr. Poonam Sethi as the resource person. And same time by 3:15 we'll be having the joining. The feedback links have been posted in YouTube as well. Yes, I am putting it here in the chat box. All of you kindly check the chat box for the feedback links. Thank you so much, Lavanya ma'am. Thank you so much, Sudhir sir. Thank you, Noor Jahan ma'am. Thank you, Ram Narayan sir. I don't know if Manoj sir is here with us now. Thank you, Shweta.
Thank you, ma'am.
Thank you so much. Participants, you can start filling the feedback from the chat boxes. The links will be opened till about 5 more minutes, and then the feedback link closes by around 5:30. So kindly have a look in the chat boxes. Even the YouTube, the chat box, the feedback link has been dropped in. I would also like to mention a special thank you to our media correspondent, Dr. Sunil sir from Veer Bahadur Singh Purvanchal University, who has been making sure that the entire coverage of our workshop is going into the papers on the day itself. In fact, yesterday's workshop details were published and shared yesterday. So sir has been working continuously to make sure that the coverage has been done properly. Thank you so much, Sunil sir. Manoj sir has joined in. Sir, आपको कुछ कहना है sir?
नहीं बस for तबीयत ठीक नहीं है. I'm not feeling well. Then
Okay,
I'll be late.
Okay, sir. So please check the chat boxes again until repeating it. The link is there. The feedback link is there in the chat box. Kindly fill in the feedback link every day for your certificates. 5:30 the link will be closed. Thank you. Thank you, ma'am.